Welcome to our webinar series for professionals.
Welcome everyone around the world, globe, in Europe, America, Asia, wherever you are. It's nice to see your faces. It's 4 o'clock here in Copenhagen, terrible rainy weather, but I'm thinking back on a very nice time I had in sunny Spain together with Dr. Enzmann, who is doing the presentation today, where I was introduced to his research in regards to CoQ10 that he will share with us today. Anyway, I am Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, a group of companies that is dedicated to change healthcare, focus on preventative strategies and treatments. And Chris was doing a town hall meeting earlier today with all the staff and employees and people that we have in the company, and he was talking about bringing in new technologies. And for sure, we will be hearing about a new technology in regards to CoQ10 today, in regards to the absorption and how absorption can be improved. So that is one of the areas that Dr. Enzmann will highlight. Remember, you have your login for your VMS account, where you can log in and look at products and lab tests. And in regards to CoQ10, I just want to mention the organics test and also the Dutch test. Mainly actually organics and metabolomics, not Dutch test. Organics and metabolomics that is looking at a CoQ10 marker on the in the citric acid cycle markers in those tests. So when you find issues there, you might want to think CoQ10, but there's going to be other issues that Dr. Enzmann will introduce us to in regards to CoQ10. So I will just do an introduction to Dr. Enzmann before I allow him to take on and share his screen with us. So Dr. Enzmann is a very young man. He was born in 1938. And he has said I shouldn't mention his age. But I have spent some days with Dr. Enzmann in Spain and listened to many of his stories in life, how he has met presidents in America and traveled around the world in a time where I wasn't even born. But he's going to allow us to peek into a snippet on his research in regards to CoQ10 that began in 1967 with Carl Volkers, and it all relates to mitochondrial health in children, their research, hypothalamus and diabetes. And he was working on a team where it resulted in the patent for thyrotropin-releasing hormone discovery with Roger Guillemin. How do you pronounce it? Enzmann and Andrew V. Shalley, and they were awarded the Nobel Prize. So this is actually nearly the closest I have been to a Nobel Prize winner, apart from His Holiness Dalai Lama that I met and shook hands with once in Klagenfurt. Anyway, they got it for their discoveries concerning the peptide hormone production of the brain. Dr. Enzmann has established the company MSE Pharmaceutica in Germany, together with his wife Uta Enzmann in Bad Hamburg in 1992. And he has also founded what's called the International Mitochondrial Medicine Association in 1998. So you are in for a treat here for the next 50 minutes, 1 hour or so. And I will pass on the stage to you, Dr. Enzmann, now. And thank you for sharing your time and knowledge with us.
Sure. Well, thank you very, very much for the opportunity to give me a chance to talk about mitochondrial medicine. Next. And I welcome you as mitochondria. And that you should do also with your patients, because then they always ask you, what is that? And then you can explain what and how important are mitochondria. Now, you mentioned already my first acquaintance with Q10 was 1967, when we tested coenzyme Q10 in children with muscular dystrophy. But the key issue is the dose we were forced to take. We had a mouse which had also Duchenne disease, and we gave 1 milligram per kilogram, no effect. 2 milligram, no effect. 5 milligram, only 10 milligram per kilogram powder is effective. This you must be aware when you treat with coenzyme Q10. For prevention, you can go to 3 milligram per kilogram. And during my lecture, I will demonstrate how we use, instead of 10 milligrams by the new technique, only 1.25 milligram. That's a new technique with nanoformulations. You see the mitochondria, and they are very active. And it's very new that we found out, or it was found out, that mitochondria can leave the cells. And we find even 3.7 million mitochondria in the blood. And within the blood, they reach the hypothalamus. And with the hypothalamus and the thyroid, they control the whole metabolism. Next page. Long ago, mitochondrial diseases were actually not realized, because 3 to 5% of are so-called by the mother. They get it by the mother. Only the mother, the woman, transfers the mitochondrial damage. But that's 3 to 5%, and most of those children die by the age of 20. What's more interesting is the secondary mitochondriopathy, which was discovered by Rolf Luft, a Swede I worked with for a long time. And we have multi-system diseases, and that means now with the mitochondria, we reach nearly all multi-system diseases. Here again, the mitochondria can be transferred out of the cells to help other cells. Next. Here, for example, you have a brain injury, and you collect mitochondria from the patient and inject it into the brain, and this will repair. The same with the heart. The next, here you see you have a heart attack, and you can inject mitochondria isolated from the patients into the heart, into the injury, and that repairs the injury. And we have done this also with stem cells. We have done studies in demonstrating if the stem cells, and especially the mitochondria of the stem cells, are very potent, they can repair even better. As I told you, what's totally new, it's maybe 2 years old, extracellular intact mitochondria up to 3.7 million. And they are the antiviral signaling proteins. That means COVID-19 really brings out the mitochondria if the virologists would understand mitochondria. Unfortunately, they don't. And here you see mitochondrial danger associated molecular patterns. You can finally see how your molecular system and immune system works. Of course, what you need is a higher level of vitamin C. You see six-fold greater risk, or even 15-fold greater risk if you have a severe before. This is what we recommend, for example, for COVID infection. Of course, ubiquinone, vitamin D3, spermidine, I come to that, selenium, zinc, vitamin C, you know, glutathione, and beta-glucan. That's a very important, I come to this issue. Just one nochmal zurück, bitte. Nein, yeah, no, next, yeah, okay. Here you see, we realized, it's long ago, maybe 30 years ago, it was discovered that antibody against hepatitis B is much better obtained if you give Q10 before. But now we found out with our fluid quinol, we treat the patient 7 days before and 7 days after, and we get very good response in hepatitis B and also with with COVID-19. Next. You see, we get titers of over 1,000, and usually you get 200. And this is very important also to prevent long COVID damage. Here you see response. If you give on the left, on the lower side, you see powder. There's a rather small response. Then we have ubiquinone drops, Sanomid is that. And then the best formulation we have in the moment, which is 8 times more effective than powder, it's ubiquinol, it's called quinomid fluid. And we have a fantastic response of 800 to 1,000 units. Now, that is a very interesting thing. That's Dunkelfeld microscopy. You see the blood is very clear, no aggregation. Now you give an injection and vaccination against COVID-19. You see the danger which is now in inflammation in the whole system. The endothelium is damaged. And now on this picture, we give quinomid fluid. You see the next picture. After 30 minutes, you have a clear picture of your blood. So this demonstrates very, very important. Any vaccination should be pre-treated with fluid and treated 7 days later, or you continuously give quinomid fluid. The same you can see in chemotherapy. Chemotherapy will also, those things. And now the newest research presented, next, you see, also this was found that you have microclots and platelets are in the blood, in the next. And this demonstrates really that how dangerous the COVID is, and also the vaccination. So mitochondria are important for the COVID vaccination and also COVID disease. Here you see, one can go on. The mitochondria like each other, like man and woman, and they fuse. And this is done to separate bad mitochondrial RNA or DNA, and then get rid of this by so-called autophagy or mitophagy. And during this procedure, you can save, for example, with coenzyme Q10, you can save healthy mitochondria. Next. Now, spermatine, probably you know about it. Spermatine was found in the semen, and it's cardioprotective, it's neuroprotective, and it stimulates the autophagy. So it's very important for COVID, and prevent long COVID damage. Next. Therefore, spermatine substitute, we have a product from spirulina, and you can apply it every day. Next. Now, what's very dangerous when you get COVID-19 is epigenetic scarring. That means if you have had diabetes and other diseases, then you have an epigenetic scarring or scaring. And there are only 2 compounds which take this away. So epigenetic means the methylation blocks the reading of the of the genome. And beta-glucan is one of the products who can rewind. So that means you reduce by giving glucan the damage by COVID-19. Here is a study we have also done. You demonstrate very nicely that you can prevent the endothelial damage. We have studied this study long ago with demonstrating that Q10 has the same effect as statins on the so-called flow-mediated vasodilatation. That means you prevent epithelial damage. That is the whole concept we have. We need energy to live. Energy means inflammation. And of course, to get energy, we need a very healthy metabolism. And we have multi-system disease. Everything is controlled by the mitochondria. And we don't talk about food supplements, which we called it, it's terrible. We call it mitoceuticals. Not, you call it nutraceuticals. We call it mitoceuticals. These are compounds. They are millions of years old. So what you have to care is they must be highly pure, and you have to do a kinetic with it and also toxicological. So we want to form a new, not pharmaceuticals and food supplements, but mitoceuticals. Next. You see the mitochondria. We don't care about the organ. We care about just one organelle. In any organ, that's a mitochondria. And this we call the chessboard or checkerboard, where you see all the indications. We have data, and where you see the mitochondria, these data are done by us, mostly double-blind studies. And I go with you some experience we have since 20 or 30 years we have brought together. Next. Here you see the respiratory chain. And here I come back to the dosing of the compounds. Now, most diseases, neurologic diseases, start with complex 1 damage. And therefore, complex 1 is reducing or activating Q10 from quinone to quinol. So for example, Parkinson has complex 1 damage. Autism children have complex 1, 65% complex 1 damage. So we need a reduced Q10, and therefore we must have a system, enzyme system in the blood, where you need enough zinc and selenium to reduce the Q10. If this is the case and you don't get an effect with Q10, you go to riboflavin. And here again, doses are 400 milligrams. The key issue, I'm coming from insulin. You cannot treat the people with 10 units. Everybody know one bride needs 120, the other patient needs 10 units. And riboflavin, we need 400 milligrams, especially if you have cardiac disease and you give a lot of Q10 and the patient cannot get up the staircase, you try riboflavin and ribose. Then, of course, we have the complex 4, which is important for the eyes, and especially for macular degeneration we can do. Now, the key issue is complex complex 5, which turns in 1 second 14 times. And for that, you need magnesium, 600 milligrams. Again, important is the dosing. And you need also creatine. Next. Now there's a little history. Kolliger found the mitochondria in the striate muscle. He's from Würzburg, then Altmann from Leipzig. He found out mitochondria originate from bacteria. So when you do wie heißt das? Antibacterial. Nee, wie heißt das? Antibiotica. If you take antibiotic, you should build up a protection with Q10. Otherwise, you damage your mitochondria. Now, what is important is that you need your whole body weight to produce per day of ATP. And this is only possible if you have enough magnesium and creatine. Now, Luft is the discovery of the name mitochondriopathy, and he discovered also the name mitochondrial medicine, and that he founded this in 1962 when he found a girl who acquired the mitochondriopathy, and she died very early, and we found mitochondria clumping together, and then of course you die. Next. This is Rolf Luft. He was for 10 years Nobel Prize President for Medicine. He was a diabetologist, endocrinologist, and I worked for many, many years together with him. This is Carl Folgers, who brought me to California, to Stanford, and he's a discovery of B12 and biotin. And here, a hint for you, you should take B12 only together with biotin to reach the very good resorption. Q10 he found 57, and together we found what was told before, TRH. We found the structure of thyrotropin releasing hormone, which regulates the pituitary and especially the thyroid. Here again, it's a total picture. We need energy, and there you have inflammation, and you get superoxide, and this can react especially in inflammation with nitrogen oxide, and then you get peroxide, and which is really powder for the mitochondria. And we have a very effective system of enzymes. The one is superoxide dismutase, and we have one mitochondrial one and two in the cells. And the mito-SOD needs especially manganium. So here again, we need really to measure all these compounds to have a good to reach a good therapy. And here again, you see the inflammation. Of course, the balance of parasympathicus and sympathicus is necessary. And what's very new, or not very new, is we found TRH not only in the hypothalamus, but also in the gut. And that is very important for our immune system. And especially the hypothalamus, as I said, regulates with the mitochondria. And as you know, 3.7 million mitochondria are flowing to the hypothalamus to regulate with the thyroid temperature and energy. And you see again mitocuticles. Next. Go back once more. TRH was found in the gut, and the reason is the hypothalamus is nothing than a burp of the gut. So we called about brain in the gut. The gut brain is very important, and it's also very important for the immune system. And you know, Pauling, he recommended vitamin C, 20 grams. And the reason, he did not know why we need TRH. All the hypothalamus need for formation vitamin C and coenzyme Q10. I come to that. Next. Now, we have a very good diagnostic system. We work here together with BioVis. I cannot go into that, would be a lecture over 4 hours, but you see, we can, and you can demonstrate on the right side, you have mitochondria. They are okay. and a few are damaged, and there are very little which have to be separated. And on the left, you have a patient. You see a lot of damaged mitochondria. And this patient cannot work, of course. And that is also the issue you get with long COVID. Next. Here again, the demonstration. Our discovery of TRH was that we imitated, the NH group is very important. If you take the acid group, you can take a kilogram, it doesn't work. TRH, you take 100 microgram. RH is very important for the fertility. Now, COVID affects the fertility of men, and you can see what they need is Q10 and vitamin C. And here I had a job with the NASA, 1967, where they want to find out the corticotropin releasing hormone for the astronauts. They wanted to prevent the stress in the astronauts. And now, we try to find TRH, which was found 5 years later. But now NASA published a very nice study demonstrating the damage of the mitochondria in the space. And here we like also to corroborate what they need is vitamin C and Q10. Here once more, it's demonstrated that TRH works in the immune system of the gut. That is a key issue, once more, Q10 and vitamin C. And unfortunately, endocrinologists and don't respect this important effect of vitamin C and Q10. Now, we found later that TRH can be used in spinal trauma, and we have several patients. They they could not walk, they cannot talk, and we were treating them with TRH and also with Q10, and they were able to walk around and to talk. So the potential also in spinal trauma is very interesting. Now here you see a picture under the microscope of coenzyme Q10, and look how powerful it looks, because this was done on the microscope. We melt Q10 and then let it crystallize. And you see, and the pathologist who did this, he was really also energized by just looking at Q10 in this picture. Next. Now, Q10, as I said before, complex 1 is necessary to transfer the inactive quinone in the quinol, and the best biphenol is quinol, Q10. And the important thing is the isoprene side chain is the most important scavenger of OH radicals, and therefore Q10 is a body-owned best biphenol we can have. Here again, we need selenium, zinc, and zinc to reactivate thyroredoxin, reductase, and the other ones. This is very important to measure. Now we come to a demonstration in 623 children. They had valve prolapse and they were actually healthy. And this was done in Japan with 620 patients. And here again, they started with 1 milligram, no effect, 2 milligram, no effect. Only at 3 milligram, and that's important, in healthy, you get an effect. And you see the steep curve. If you go with a Q10 a little bit, and don't say take a little bit Q10. No, you must reach up to the saturation curve, because if you go a little bit left, you lose immediately energy. And 50 to 60% of energy is used for regulation of all channels, calcium channel, chloride channel, and natrium and potassium channels. Next. Now look at the mitochondria. They have a diameter of about 500 nanometer. And then we went to discover nanoquinone, which has a diameter of 50 nanogram. Take the next. Here you see a picture of sanomid, that's sanomid, but quinoid fluid looks the same. And we have within you have pure Q10 melt. So it's a cold melt, and without you have some lecithin and some water and glycerin. That's all. But we have and here you see the different resorption rate. Powder has about 3 to 5%. Sanomid has 40%, and of course quinoid fluid, as I said, you had 8%. And if you calculate a patient is 80 kilograms heavy, we need times 10, 800 milligrams. You cannot eat it and you cannot pay for it. Therefore, with fluid, you need just 100 milligrams. And the next shows it's very fast. And we just have done in Sevilla a study with Navas and Lopez, and comparing, for example, Pharma Nord's product and the Red Hour product, Sanomid, and you see very clearly the best formulation we have in the moment is quinoid fluid. And you see also how fast it works. Therefore, it's not only for prevention, it's also in intensive care. Here you have a patient which regulates, well, not so good, but 6 is okay. But as you see, we measure 5 minutes. It's a heart rate variability. You see the heart, the yellow line here is very weak, and you had arrhythmia. And this was done, I cannot read, 12 o'clock 25. And now we give 20 drops of Sanomid, and half an hour later, you see very nicely, the heart has very power, the arrhythmia are gone, and the regulation is again very, very fast. So this demonstrates really you should have this fluid always with you. Again, once more, the chessboard. Next. Now I go with you a few indications, and cardiology was done the most. Heart failure, for example, and this demonstrates very nicely this. These are over 400 patients done by Löngchen. Pete Löngchen, I know very well, also worked with him. These are over 400 patients in class, New York Class Heart Association classification, and you see 3 patients cannot work. Sorry, three patients cannot work in stadium 1, 250, 230 cannot work, and they cannot even get up 50. And on the other side, 3 months after Q10, and mostly only 1. On the left side, we have 5 to 6 medications. On the left, maybe 1. And you see the steel drop. You have now 212 patients being able to work fully. And I would recommend really in the work, the doctors who are working, what's their name, as medical doctors in companies, they should really recommend for their people to use Q10. Next. The study by Mortensen is one of the biggest studies. When I was also within the group of Q10 Association, which I founded also with Mortensen, we recommended this study. And I recommended the fluid, but they took the pharma-not formulation because it was a capsule and the fluid was too complicated for them. But the results are fantastic. As you see, you just take 50% of cardiovascular events, mortality, and total mortality. And the problem is cardiologists don't care about that. It's unbelievable. Next. Now, this is our study. Now, Damien is a neurologist. He was at the University of Dresden, and he was always called by the cardiosurgery. And when people came in with after heart arrest, and he was embarrassed because the heart was beating, but the brain was gone. And therefore, they had done some experimental data that Q10 is very neuroprotective. And this study was done by two lady doctors. They wrote their thesis on that. They gave 250 milligram when the patient came in, and 3 times for 5 days, they got 150 milligram. And the outcomes you see, whereas the normal treatment leads that to 29% survival after 3 months. And with our treatment, once more back, it's 68%. And you see how powerful this treatment is. You see in the next graph. This you see very seldom in drug research. Then you need 1,000 patients, and here you see it very, very effectively, and especially these patients survive very nicely. Next, and also very important is now also the brain, the blood barrier. Blood barrier is closed during this treatment. And this is, for example, the reasons that the American soldiers get coenzyme Q10 after they came back and with brain damage, usually, or brain fog. And the reason was their blood-brain barrier was open. And of course, during the war, they got damaged. And you see nicely, Q10 can help to close the brain damage, the blood-brain damage. You see nicely how it is, and also the so-called, I forget what it is. What takes the next? And you see, whereas here in Germany, we don't get any respect for those studies. We got into the guidelines of the American Neurology, reducing brain injury following cardiopulmonary resuscitation. And you see how powerful Q10 is working in the mitochondria. We did another study with Professor Hescheler. We take cardiocytes and treat them with hydrogen peroxide, 30%, and you see you get arrhythmia. If you treat after or before, you don't get arrhythmia. So Q10 is very important with magnesium, of course, selenium, and other compounds to prevent arrhythmia. The diabetology that I worked with Rolf Luft. Next. And you see, statins are reducing Q10 synthesis, but you need Q10 to form insulin. So if you get a statin, you can be aware to get diabetes too. So again, a statin should be always combined with coenzyme Q10. Next. Here again, you see the increase of diabetes 2 in the meridian of introduction of statins. Next. Eyes, glaucoma, AMD. Here you see especially the main cause for is AMD, and what you see is that there is a compound which blocks the cytochrome C, and if you blocks the cytochrome, the complex 4, cytochrome C goes into the cell, and you get apoptosis. But we can prevent this with negatively charged cardiolipin or with negatively charged ubiquinone Q10. So you can there we ourselves don't have a study yet, but there is a study already. Next. You see the positive, it's like a whale in the eye you cannot see, and then this positive is taken away by the negative loaded Q10 particle, and you can see again. Of course, not everybody, but you can try. Dentistry, very important. Dental health is very important. Here we did a study long ago demonstrating that gingiva and also parodontitis can be prevented. Next. So you see the blood circulation and energy supply of the mucosa, compensation, bioenergetic deficit by Q10, free radicals, reduction of bleeding density, plaque and pockets, and of course wound healing is better, and firm, healthier gums. Next. That is also a study which is important for Alzheimer. Alzheimer was for many years, we thought we need an antibody to beta amyloid formation. And that was totally wrong. Beta amyloid is an body-owned antibacterial compound. So what you have to test, and every doctor can do this by taking some sulcus and send it to a laboratory, and they can find out if you have this pyroformanus gingivalis. This can induce a lot of beta amyloid because, of course, the brain cells want to destroy this pyroformanus gingivalis and produce a lot of beta amyloid. But by that, of course, they kill themselves. Next, oncology. Next. Oncology is quite interesting. When a baby in the first 2-3 months, the mitochondria are inactivated, and only we need growth. But then at a certain stage, the mitochondria start to work, and then you can control. And this we do in cancer. We must just turn it around. So we have to induce mitochondria to work, and that gives cancer cells into apoptosis. Now, this is a new and fantastic finding. Unfortunately, very, very bad for us. You see here on the left picture, you see on the right side a cancer cell. On the other side, you see an immune cell. And now the cancer cell builds a tunnel towards the immune cell and takes the mitochondria out. Now, how can this immune cell work against any virus? That's unbelievable, which was discovered only now. It's 21, I think it was published. But that is, now, what can we do? We don't know yet. Next. In cancer, when you have some X-ray or radiation, you can protect your gut very nicely by giving Q10 and especially ubiquinone. Next. Of course, tinnitus. Tinnitus is also a mitochondrial problem. We have done studies. Next. And you can demonstrate very nicely. You can reduce tinnitus, and even when you have hearing loss, you can try to help the mitochondria to get some of those tinnitus patients back to normal. And you see very nicely in this patient, that was an engineer. His wife gave him one day, he had tinnitus for many years, and treatment didn't work. But then his wife gave him some Sanomid, and then he realized, oh, I don't hear anything anymore. And he said, oh, I gave you something. Of course, engineer starts again, and you see nicely, you need Q10 all the time. Of course, if the stress is lower, you can use the dose. But you see nicely, it works. And that is very impressive. Mitochondrial medicine works, and you can really demonstrate it. Next. Neurology, especially this we started at the University of Dresden, and you see it, especially here, the glutamate exitoxicity can be also reduced by Q10. It's newest work. The damage was thought of the brain is especially glutamate exitoxicity. We demonstrated in the study with Damien, we can reduce the glutamate exitoxicity. And the reason for the damage is the membrane potential is losing. And we can build it up with Q10. And of course, other compounds. Next. Here you see stroke patients, survival, and the controls. You see, this is Q10, very low, and these are the stroke patients. And SOD, you need especially ND, and you see a very nice effect on the calcium overload. Next. Here once more, you see the effect. The calcium overload is controlled. Another disease which is coming up more and more, if you like, especially strawberries from China, and you have nice, we have this pesticides, then you observe this disease. That's a progressive supranuclear palsy. It's a palsy of the eyes, and the patient cannot look up and down cannot turn the eyes up and down. They present with instability, they fall, they have frontal dementia, we demonstrated in the brain, and they have dysphagia, and they die within 5 to 6 years. So that is a disease you will see more frequently. And you see this, it's not, you think maybe it's Parkinson, it's not. It comes up with like Parkinson, but dopamine will not, or not dopamine, will not work. And we did a study here at the University of Marburg, and you see only after 6 weeks we have safe and improved cerebral energy methods we demonstrated by MRT measurements. Now psychiatry, now we are back with the children, especially with autism. And autism is based on the mirror neurons in the brain. They cannot communicate. They have too little energy. And when you give, especially Q10 and other compounds, they can compare. So they can look at you, they can talk to you, and we do just a double-blind study, but we have many patients. They are very nice in the school, in their home, and also in kindergarten. So the reason is also, it's a complex one, damage in 64 patients, in 64 of the patients, these poor children are in the wrong hands. They should be treated by a doctor experienced in mitochondrial medicine. Pain is also, pain needs energy. And of course, pain, especially or admission is migraine, especially produced in the hypothalamus. Here again, my nice organ, and you can nicely treat much better and without any side effects with Q10, magnesium, and other substances. Next, please. First, it was demonstrated it has a reduced mitochondrial energy reserve in the brain. So if you replace it, then you should get an effect. Next picture. You see nicely energy defects, and they are gone when you treat with Q10. First, that is our study. And you see we have a gain of nearly 30%, 60%. So we have a very good effect on complex 1, 2, and 3. Therefore, we combine usually B3, vitamin B3, B2, and Q10. In addition, we give magnesium. Next. And you see, we are in the same group with other. I would not give a child a beta blocker for migraine. We could easily get magnesium, riboflavin, coenzyme Q, or we have a formulation which is Ripomid, no? What's his name? Okay, I come close to the end. Now we have here a traumatic brain injury, and it's one case report, but we have 22 cases with the same result. Next. So the patient was falling back the staircase, and next, please. She had intracerebral supradural brain damage. The shift of the left brain, you see here, the middle line of the brain is pushed to the right, and also the ventricle is closed. And this was, of course, a very bad situation, and there was very little hope that the patient would survive. And I was driving with the patient and asked the doctors, you should take quinoid fluid. They said, take your wife, that was my wife, take your wife we don't do that. I said, I bring the decision of my wife, we begin with the treatment. And then we were starting treatment, and you see 24 hours later, the middle line was again here. Of course, we had to take the skull also away, but here again, Q10 also can help tremendously because you take the edema away. Q10 regulates the water channels, the aquaporins, and they are very important in the eyes and the edema. And so always think about if edema, you should use Q10 and the fluid Q10. Next. Now you see mitochondria activates mitochondria of all organs. This results in an extraordinary preventive and therapeutic potential, especially in the area of multisystem, but also in intensive medicine. This demonstrates this is a mitochondria. We must reach the mitochondria. Yeah, not outside, but we need Q10 and vitamin D3 in the mitochondria. And you see here, this is the mitochondrial Eve from Africa. Everybody of us, we come from this girl in Africa, and because the mitochondria are given further to the children, but nothing from the mitochondrial code of the man. So that's very important. Next, finally. Well, thank you. So thank you very much for listening. Of course, you may have many questions, and usually I need 2 days to explain it. But thank you for listening, and I hope you can start on Monday with mitochondrial medicine. All the best. Thank you very much.
Thank you, Dr. Enzmann. Thank you very much. As I just wanted to share Dr. Enzmann with all of you when I met him in Spain in November, and I was like, his knowledge is so important to all of us. And it is a gift, hopefully, that you all feel the same, to have spent this hour with Dr. Enzmann and him explaining all his amazing work in regards to CoQ10 and health, you can say. Some questions have come up, and what's very, very important before we close this meeting, so I'm going to take that as the first one, is, and Dr. Enzmann, you have to help me, because you know I'm not the best mathematician. You have to help us all with how do we calculate the dosage, and is it the same dosage for most of the conditions? So, but let's start with the dosage for someone who weighs 80 kilos or 70 kilos. What should we start with? In the drops, we have the drops here.
Okay, once more. For treatment, if you have a disease and you take the patient's weight, I take always 80 weights because it's easy to calculate. So you need 10 milligrams. So 10 times 80 is 800 milligrams for powder.
For powder. But then because we are in this
you reduce by 8. We need 1/8. It's 100 milligrams.
So that's 100 milligrams.
And in 1 drop is 6.5 milligrams.
And 5 drops is 32.5.
Yeah.
So we move
10,
up
12,
roughly
10 to 12.
Yeah.
And we start usually 3 times 10 drops, but if you calculate the basis we found out in California, then you take these 100 milligrams divided by 1 push, 6.5, and that's about 12 drops, or 3 times. But I would always divide it up 3 times if possible.
So you get a stable, steady state in the body.
Right.
So a lot of diagnosis, atrial fibrillation is coming up, migraine came up, fertility came up.
Yeah.
But mitochondria is important for everything.
Yeah, exactly.
So if there is a condition where you suspect the mitochondria deficiency, you go for this dosage.
I go, exactly.
If you have a healthy person that is full of energy,
3 milligrams.
Nothing, then it's
3
milligrams.
3 milligrams. Take it by 80 kilograms. It's 240 milligrams divided by divided by 8. What's it said? How much is it? Divided by 8. It's usually 3 times 3, 3 times 5. And that's important also for children. You do the same. Yeah, what we have done in California with those dystrophy patients.
So there you go. What
you can do, because the half-life of Q10 is 30 hours. And usually it takes 10 half-lives to get rid of all Q10. That means 10 times 30 is 300 hours divided by 24. It's about 12 days. Yeah. So you can reduce, if you see an effect, usually see an effect in in 2-3 weeks. Then you can reduce of the half. You can go half. Yeah. But you must measure once in a while because the situation can be worse again, then you go up again. If you have additional stress, if you have surgery, yeah, then I would go up again. So flexible. That's like insulin. When I run around, I need less insulin. If I'm sitting around, I need more insulin.
Super. So I'm gonna just quickly look at my notes here. What else I had? What drugs do we need to be aware of? There is, of course, statins and CoQ10, but is there other antipsychotics, antidepressants, other medications that? Actually, what we must request, if you register a drug, how it damages the mitochondria, or takes, not damage directly, but takes away the compounds you need for your health. For example, what it's called, the proton pump inhibitors, PP, they are very bad. They are very bad. I was head of research when we introduced it, and I knew the toxicity, and I saw tremendous cancer in the stomach. Yeah, so you can take it 4 to 6 weeks, but not longer. And I requested actually that it should be given only by the doctor and not. And now it's free on sales. It's unbelievable. And we have developed compounds which we call food supplement. Nee, wie heißen das? Nutritional
compromise. Wie heißen die? Wie heißen die da hinten? These are Arzneimittel-Ergänzungsmittel. That means if statin takes away Q10, you give Q10. If you have especially the proton pump inhibitors, there you need especially B12 and the others. But yeah, this we have also a formulation. The pill is also very bad for girls and women, very bad. And we have also metformin is a mitochondrial poison, but it's #1 in the guidelines of guideline #1. The diabetes is terrible. So most drugs have an effect on compounds which are necessary for the mitochondria to work. This you must be aware.
Thank you, Dr. Enzmann, for sharing that. That is very important information, and it is also very brave saying it. But of course, this group understands exactly where you are, where your point is in regards to medication. And of course, the aim of our treatment is to make people medicine-free.
Yeah.
So that we can fix the mitochondria. But even you mentioned antibiotics as well during your talk, etc. So I am going to finish off now, just with a few notes before I say thank you to you and thank you to everyone else as well. So we will share the recording with everyone. We will share the slides, and I will put the calculation into the mail where we share these things with you, so that you have a clinical tool for Monday, as you said, or tomorrow. So thank you, Dr. Enzmann, for your time. It was a gift to listen to your presentation. I think there is a lot of things to revisit in your talk. I have just want to say one thing before I let you say the final goodbye to everyone, that the product is in stock, but right now, if you're checking it, there seems to be a glitch, but it is there. So tomorrow it will be there for you. It is in stock. So I think we just need to press one button and then it's live. So anyway, Dr. Enzmann, thank you for your time. Enjoy your evening.
Thank you. Thank you very much. You could do me a favor.
Yes.
Some criticism, write down some, so I can improve myself.
Oh, no, you're perfect. You're a gemstone as you are.
No, sometimes.
I think you have something, though. I think you have something hidden behind the camera. I think you have a flag.
Yeah, yeah. Denmark, yeah.
Yes. I noticed it. I don't have a flag. I'm so sorry.
Okay. Thank you very much.
I hope to
say hello.
Everyone, take care and look out for your email and hopefully see you soon. Take care. Bye.
And if there are questions, please write.
I have your email, so I will forward the question.
Okay. See you. Bye-bye.
Bye. Bye, everyone.
Good.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.